So welcome everyone. It's really a big pleasure to be gathered today for this first day lunch talks. So it's a webinar series that is co-organized by the POPR Biotherapie, but also the Réseau des Intégrateurs and France Biolide. And this series of webinar will be held once per month. So it will be the first day of each month at 12. We try to focus on a short format, so it won't last more than 45 minutes. And the idea is clearly to provide you with an update of the latest innovation in the field of cell therapy, gene therapy, and bioproduction. So this will be the first meeting, March 6th. So you can already note on your agenda, we will have a presentation regarding the integrator perspective, and April 3rd, it will be more about an example of innovation regarding the strategy for quality control evaluation, and it will be from clean cells. So we will provide you with the calendar and with when it will be totally done. And again, thank you for being here. So today it's the bad example because we are facing this technical issue, but normally the seminar won't last more than 1 hour.
Cécile, you can move forward with the introduction. We have the file we can share.
Okay, but go ahead, Fanny. Go ahead.
So I will go ahead with the introduction. So first, thank you all for joining us today for this first webinar of the series, of the Thursday Lunch Talk series, dedicated to gene therapy. So today we are honored to host Dr. Christophe Mulle, who is a research director at the CNRS Interdisciplinary Institute for Neuroscience at the University of Bordeaux. Dr. Mulle is a cellular neurobiologist specializing in the electrophysiology of synaptic transmission. He is recognized for his pioneering research on glutamate receptors and hippocampal synaptic plasticity. His groundbreaking contribution to the study of neurotransmitter receptors and synaptic circuits have been widely acknowledged, including by the Grand Prix La Monica de Neurologie from the French Academy of Science in 2014. In addition to his fundamental research, Dr. Mulle has been actively involved in a translational project targeting aberrantly expressed kainate receptors in temporal lobe epilepsy, in close collaboration with Dr. Valérie Crépel from INSERM Marseille. This important work will be the focus of today's presentation. We encourage you to take full advantage of this opportunity and engage discussion and ask questions. So please join me in welcoming Dr. Christophe Mulle.
Hello, everybody. Thank you for inviting me to present this work. I'll try to be not too long. I'm not sure. I don't know exactly the background and everybody, but you will tell me if things are not crystal clear in terms of the neuroscience behind the work that I am going to present you. So indeed, this is a part of the work of the lab, and it's really a very strong collaboration that we have had with the Valérie Crépel from INSERM in Marseille since more than 10 years now. And it started really with a fundamental discovery of a mechanism which, with a lot of good stars around this project, went from this basic finding to something which is really advanced in terms of clinical trials now. Can I have the next slide? Because I'm not in control. I'm sorry about this. It's a bit complicated. I'm not in control of the screen. So just a very quickly disclosure that we have been, Valérie and myself, founders of a company, which was then sold to a bigger company, Unicure, and we have been consultants for this company. The next slide. So the project is about kainate receptors, and kainate receptors are one of the family of ionotropic glutamate receptors. So for those who may not know, glutamate receptors are the key receptor channels which are involved in synaptic transmission, excitatory synaptic transmission. So you have release of glutamate from the synapses and then activation of different types of glutamate receptors. And kainate receptors are one of these three. The two main other ones are NMDA receptors and AMPA receptors, which are much more studied and much more abundant in synapses than kainate receptors. So kainate receptors are a family of 5 genes, glucase 1, 2, 3, 4, 5. It's simple. And in fact, there are tetrameric receptors which can combine different subunits. And the major subunit in the brain is the subunit combining glucose 2 and glucose 5. So 2 glucose 2 subunits and 2 glucose 5 subunits, it forms the receptors, which when glutamate binds, opens a channel and lets cations to go through and leading to depolarization of the neuronal membrane. Next slide, please. So there is a large diversity of physiological roles for kainate receptors. If you look at the top panel, I show, in fact, in this review that we wrote recently, that kainate receptors that can exert their role in many different ways, either at the presynaptic level, so this is number 3 and number 4, in the regulation of the vesicle of glutamate, the release of the vesicle of glutamate, or at the postsynaptic level, 1 and 2, and acting principally as an inotropic receptors, meaning that when you open the channel, you have cations that flux through the channel, and they have also been discovered to activate an indirect pathway through a metabotropic mechanism and regulate potassium channels. In general, the activation of these receptors are very important for the regulation of the activity of circuits in many brain areas, and we in the lab work on hippocampal circuits. What I wanted to show you, and I don't have anything to point really, but if you look at the left part of the panel below, so you see that some synapses from DG cells to CA3 pyramidal cells, they are very abundantly expressing kainate receptors, but others are not. And this creates a very specific role for the kainate receptors in regulating the activity of neural circuits. And I will show you, and this is the line in the line going from DG cells to DG cells, that in the condition of temporal lobe epilepsy, there is a recurrent connections that is made from DG cells to DG cells. This is a pathological connection. And in fact, we have discovered, this was the starting point, that these connections are using kainate receptors. So next slide, please. So now temporal lobe epilepsy, this is really our target pathology that we've been working on. So again, so it's a chronic disease. It's characterized by unprovoked recurrent seizures, which are a major activation of neuronal circuits that create very strong phenotypes, symptoms in the patient. The most common forms of epilepsy have focal seizures. It means that it starts from a very specific region and then can spread to other areas of the brain. And in temporal lobe epilepsy, this focal point of the starting point of the discharge is principally in the hippocampus, but also in other parahippocampal areas and in the amygdala. So next slide, please. So yes, so this is our target is mesial temporal lobe epilepsy, meaning starting from the hippocampus or amygdala. And in fact, what we know is that there are diversity of possible causes, starting with head injury or stroke, brain infections, lesions, brain tumors, and in a small number of cases, you have also genetic syndromes that can cause this mesial temporal lobe epilepsy. What is important to state here is that the trigger can be occurring very early during the childhood, and then it is followed by the latency period, which can be quite long for several years. And this is during this period that you have a reorganization of neuronal circuits in these brain areas which have been affected by this initial event. Very often it is accompanied with a hippocampal sclerosis, so some cell populations decrease and die. And very importantly, not only do you have seizures, but you also have a lot of comorbidities which are linked with a dysfunction of the hippocampal circuits. That includes memory, because the hippocampus is a major area for encoding new memories, for instance, for spatial navigation, and also it also plays a quite big role in anxiety and depression. So because the circuits are not functioning any longer in the right way, you have these comorbidities which are very difficult to treat. Next slide. So how can we treat temporal lobe epilepsy? So there are many symptomatic treatments available, which are these anti-epileptic drugs. And this is the primary treatment, that it works well in many cases, as you will see. So what it leads to is a general decrease of the excitability of neural circuits. So you need to keep neural circuits alive, but you want them avoid the circuits to be completely dysfunctional and create this very large synchronous activity of neurons that create the seizure. So the targets for these are GABA neurons, GABA receptors, ion channels, release of GABA, also glutamate receptors in a way. And these prevent, so this is a symptomatic treatment which works well. And as you see in the diagram below, you have about 70%, 65% of efficacy of the pharmacological treatments in patients with one or a second anti-epileptic drug. But you see that in blue, in dark blue, that there is a lot of many patients are not sensitive, cannot be treated with this. Next slide, please. So what happens for those who after two treatments phase with the anti-epileptic drugs, what can be the way to treat these patients is surgical resection. And what it means is that you have to localize the epileptic focus with an electrode that goes down to localize in the hippocampus, for instance. And then there is a resection of this part, but when you do the resection of the surgical resection of this hippocampus or amygdala, you also remove part of the temporal lobe, which is above these structures. And indeed, it works quite well. It leads to seizure freedom, and for about 80% of the people. There are some risks. The risks are, of course, those linked to surgical procedure, and also a number of post-operative cognitive deficits. And for many reasons, there are only a small number of patients which are eligible for surgical resection. So there is a real, next slide, please. There is a real need to find a new way, a new treatment, because these surgical resections are not possible for many patients. So we have been working on an animal model of temporal lobe epilepsy, and these animal models reproduce the large electrophysiological discharges that you can see, ictal activity on the right. So what you do is that you inject pilocarpine to a mouse, creates acute seizures, or a rat here in the case, it creates an acute seizure, and then after a latent period of several weeks, there is you start having unprovoked chronic seizures. And you can record these by putting a radio frequency transmitter in the hippocampus, and you can have recording over over several days, as you can see below. So each 1, 2, 3, 4, 5 means different types, different animals, which have, and you can see on the right the little bars, which are the seizures. So it's variable from one animal to the other, but you see that you can have up to, let's say, 10 seizures a day for these animals that you can record. So next slide. So the basic thing is that I already alluded to. The basic mechanism that we have found is that, so here you have the dentate gyrus, this structure in the hippocampus that sends an excitatory transmission to the stratum lucidum in CA3. So another cell, so these are excitatory synapses. And these cells, these synapses, they use kinetoreceptors. But in the context of temporal lobe epilepsy, there are also a reorganization of circuits that we call pathological sprouting of mossy fibers. And these cells, they make synaptic contacts. These axons make synaptic contacts onto themselves. And what Valérie has found 20 years ago now is that these fibers, these synapses, they use kinetoreceptors. And we have further defined which kinetoreceptors these were. And we found that glucate2, glucate5 heteromeric receptors were the receptors which were involved in this recurrent activation of the dentate gyrus through the recruitment of aberrant kinetoreceptors. Next slide. So very quickly, just a summary of what we have found is that, so using a rodent model of temporal lobe epilepsy, we have used the glucate2 knockout mice, and we have shown that glucate2 knockout mice are also glucate5 knockout mice. Well, when glucate2 is not present, glucate5 is not present any longer. And what we found is that in these mice, or using a glucate2, glucate5 antagonist, we have a major reduction of seizures, as measured by through telemetric recordings. So this was a starting point. Next slide. So this was a starting point of our translational project. So really, we have aberrant kinetoreceptors, which are a target for temporal lobe epilepsy. And at this point, we had the choice of looking into different things. We started to collaborate with chemists to design good glucate2, glucate5 antagonists. We started also to use a peptide approach to try to prevent the recruitment of kinetoreceptors at synapses, at these aberrant synapses, but mainly together with the SATT Aquitaine and with a fund venture capitalist, Kurma, we decided to really focus on gene therapy. And the idea of the gene therapy is to inhibit the expression of glucate2, glucate5 in the hippocampus of the patients with temporal lobe epilepsy. This led to, after a few years, to the creation of a company, Corliss Therapeutics, for which we were founder with Valérie Crépel. And then a couple of years later, this company was sold to a bigger gene therapy company, which is called Unicure, which is in the Netherlands, and which is carrying on the project. So that's about the structure, general structure, but I will show you what we have done. Next slide, please. So just to come back, gene therapy targeting kinetoreceptors in temporal lobe epilepsy. First, it really is an important important unmet medical need for drug-resistant temporal lobe epilepsy. As I mentioned, about 30% or more of patients with temporal lobe epilepsy are not cured with the cannot be treated with the anti-epileptic drugs. We have, I think, discovered that the target and mechanisms are well characterized in preclinical models, and I don't have time to show, but we also have good evidence that this is the case also in the in human samples and brain tissues from temporal lobe epilepsy. The idea is to use a viral vector for the expression of interferent RNA against GRIC2, which is a gene encoding glucate2, to downsize the expression of glucate2. And an important thing is that we plan to have a local delivery of this viral vector, in a subpopulation in the hippocampus, where we know that the focus of the discharge, the epileptic discharge, will be. In fact, the surgeon, when they try to localize the focus of the discharge, they already go down with an electrode to a stereotaxic electrode to localize this. So they have already the track to go down in the local place where the virus can be delivered. And as I will show you that we can test this in human, we have tested this in human tissue cultures. Next slide, please. So this is the basic outline of the work. So we target GluK2, Greek2, the gene Greek2. We use a neuronal promoter to express a microRNA against Greek2. And we use AAV9 vector, which is very neurotropic. And as I said, the idea is to do local injection of the AAV vector in drug-resistant patients with TLE, where we have the focus of the epileptic discharge have been already characterized with the stereo EEG recordings. Next slide. So the first thing that we did was to test the efficacy of gene therapy vector in a mouse model of temporal lobe epilepsy. As I said, so you inject pilocarpine, you wait 8 weeks for the time for the circuits to reorganize and become really epileptic. Then we do a viral transduction into the local infection of the virus into the DG. And 2 weeks later, we start looking at the different aspects of epilepsy in the mouse. One key aspect of epilepsy in the mouse is that the mice become extremely hyperactive, and this is easy to look at in an open field. You can automatically look at the activity, locomotor activity of the mice, and you see that they start increasing a lot, doubling about their activity. And then from 8 weeks to 7 weeks, even longer, we use telemetric hippocampal EEG recordings. So the first thing that you can see on the bottom graph is that we have a reduction of the GluK2 protein in the hippocampus, but it's not major. It's around between 30 and 50%. So we don't remove all the kinetoreceptors, which are expressed in the hippocampal neurons. What we see is that on the next graph that there is the injection with this mRNA anti-GluK2 viral vector leads to a restoring of the locomotor activity. But most importantly, as you see on the right graph, the number of seizures per day drops to a very small number of seizures. So it's extremely efficient, even though we just remove 30 to 40% of kinetoreceptors. We have a major effect on seizures, and we interpret this as the fact that in fact kinetoreceptors, by removing kinetoreceptors, we go below a threshold for excitability of the circuits. I mean, we can explain that pretty well by looking at the synaptic connectivity and the excitation of neurons in the hippocampus. Next slide, please. So also, which is quite nice, is that by using these AAV vectors, I'm not going to go into the details of the protocols, but we can show that there is a restoration of. So let's see. We have a setup to look at anxiety using a light dark box apparatus. You see that the mouse which is anxious will not go through the door, through the light, but so you see that the epileptic mouse, on the graph in gray, goes very little to the light box, and then you recover statistically this transition from light to dark box, from dark to light box in the epilepsy with using the viral vector. We also show that we have an improvement of working in spatial memory in these mice, and that you can see that on the graph below right. Next slide. And so in fact we can show that using these viral vectors in the preclinical model in a mouse, we improve not only seizures but also all comorbidities which are linked to this disease. But also what is quite interesting and is that we have been able to use tissue from epileptic patients. So we in Bordeaux and in La Timone in Marseille, we have been using a hippocampal resection from patients with temporal lobe epilepsy. So we get the hippocampus from these patients, and then we can slice it and put it in a culture dish, and we can keep the slice for over 2 to 3 weeks. And at the next day after putting the slice in culture, we can infect or transduce the hippocampal neurons with the AAV9 vector, and you see that in the below. On the right, for instance, you see that all the dots are neurons which are transduced with the AAV9 vector, here in that case expressing GFP under the control of the synapsin promoter, and only neurons are transduced. Next slide. So then what we can do is then look at the effect of our gene therapy vector on first on the level of kinase receptors in the hippocampus of patients. And again, we see, this is on the bottom right, that so we have a good transduction with our vector of the neurons. We have a pretty high level of neurons which are transduced. And you see on the bottom right, on average, we have a 30 to 40% decrease in the amount of the protein in these slice cultures. Next slide. But most importantly, what is important is to look at what it does in terms of discharge, epileptic discharges. So here we cannot really have the epileptic discharge. I mean, it's a bit complicated to go into the details, but what we have, these are called they are called interectal discharges, and you can see that you have a major reduction in the frequency of these interectal discharges, as you can see on the right, using our AAV9 vector for downregulation of GluK2 in the hippocampal slices. So we have a good indication that in the hippocampus human hippocampus from patients with the temporal lobe epilepsy, this AAV vector works. Next slide. So here, in summary, together with the company Unicure, we have created a clinical grade A vector for gene therapy by targeting the GRIG2. So here you see on the little diagram that in fact we have been using two microRNAs in tandem. And then we do local administration. We have been doing local administration in the hippocampus of epileptic mice, or also in the non-human primate, to see that we have a really localized expression of this vector. And in the mouse, in the epileptic mouse, we have shown that it decreases dramatically the number of seizures. It improves the health. I haven't shown you this. It decreases anxiety and increases cognition, stabilizes cognition. So this is all good, in fact, at the moment. And the next slide, I don't remember what we. So yes, so we are not involved anymore in the project since the company was bought by Unicure. But in September 2023, the Unicure has announced that there was FDA clearance to investigate this drug. So the drug that is called AMT-260 now. It's a gene therapy vector for temporal lobe epilepsy. And we were very happy to hear last November that the first clinical trial had started. The first patient has been dosed in November 24 to start a trial of 12 patients in the US treated with this AAV vector. Next slide. And these are the people who mainly did the work in Bordeaux and Marseille, and with Unicure, and also in collaboration with Regenex Bio for the vectors, the AAV9 vectors. And I thank you for your attention.
Thank you so much for the presentation, actually, for showing all those great results. And honestly, it's also great. I mean, it's also really great to see that you are now starting the clinical trials. I mean, you are the company, of course, who is taking over the development. I wanted just to say to all the people connected that you could ask questions if you go to the question and answer session in Teams. You could write actually your questions, and we will ask them to Dr. Mulle. So please don't be shy, not in the conversation, but in the Q&A icon that is on the top of your screen. Maybe may I start with one general question? I mean, you have been involved from the first discoveries up to the creation of a company, and then I guess that even if you're not involved yourself directly, you are still following the development. What were the main obstacles that you have faced from when obtaining the first proof of concept to getting through the first steps of the clinical translations? And are there some lessons learned that you would like to share with the academic people who are looking forward to make this step?
I think that I mean, we really started with a basic mechanism mindset. I mean, we did not want to cure epilepsy to start with. I mean, we really wanted to understand the mechanisms. And I think that one important lesson I would like to convey is that it's very important to have very precise mechanisms in hands to understand what why it works in the later steps. So I think that we were lucky to be able to go far enough into the mechanisms because of our expertise, Valérie and myself, for sure, but also because we had a support from 2 ANR grants that allowed us to really go into the details of the mechanisms. When you start working with a company, then the mechanisms are not really at the same level of I would say, importance. They want to move forward with a medicine or something, whatever you call it. So it's important to really to have very strong results in terms of basic research.
And I guess also that, I mean, this better understanding of the mechanism is also helping really to focus on the clinical application, on the type also of the administration and all details that then would be absolutely crucial for the development.
And then I would say that we have been also very lucky because we met people. I mean, this venture capitalist Kurma and the SAT Aquitaine here, who believed in our project and in some transfer as well. So it was, I mean, we learned a lot of things, and I would say that in some transfer for all the patents that they have helped us file, etc. I mean, it's a lot of things that you learn as when you are a basic scientist, but it's a lot of fun.
Thank you again for your presentation. There are 2 questions that have been raised. One from Angali, who is asking if there are any evidence of immune response to the AAV in the hippocampus. And what were the regulatory authorities' question on this aspect?
So so this work, which we didn't do, I mean, it was the company, we did it with the CRO, looked at the expression of the vector, the presence of the vector in many brain and organs, brain areas and organs, and it was considered to be very low. Something that we don't really know is whether there is expression in whether it triggers an inflammatory response. But as I showed, we are using a neuronal promoter, so we have no expression of our construct in human cells in the brain. That's for sure. And the other thing is that that's. And we don't see overt signs of neuroinflammation when we do local injections. I don't know if it really answers the question, but I would like to say something about this point. So we know that there is no expression of our microRNA in immune cells of the brain because we use a neuronal promoter, but we don't know whether these immune cells are not transduced by the viral vector. That's something that we don't know.
Thank you so much. And then there is also another question from Philippe Vernier, who is first congratulating you for the talk and also send you a friendly hello. And
hello.
Then it's asking, and then he's asking. I understand that the occurrence of recurrent axonal collaterals in DG cells plays a major role in the creation of the epileptic condition. But does the expression of the GluK2 and 5 but does the expression of GluK2 and 5 also announced or spatially modify DG cells as a condition for seizure to occur?
So I think that so the question is about the properties of DG cells in the context of temporal lobe epilepsy. When we have so the morphology of the cells don't really change themselves, except that they send axons to themselves. Okay, that's a problem, and I think this is linked to the disappearance of another population of neurons, which are called MOSI cells, and normally, these MOSI cells, they receive axons from synaptic contacts from DG cells, but since they are not there any longer, it seems that the connections are made within dentate granule cells. So, I mean, it's a reorganization of the circuit which leads to this aspect. I would say that there is also something important is that we don't have necessarily an increase of the expression of GluK2 or GluK2, GluK5 in these cells. What we have is a reorganization of the receptors. I mean, they are positioned at a place where they should not be. So it's really yeah. I hope. Not sure I understood. I answered the question.
Yeah, it was wondering if this was the main rationale for reducing the GluK2 expression in the treatment.
Yeah, this is absolutely the rationale. I mean, it's because, as you can understand, if you have an excitatory synapse onto an excitatory neuron, it makes a very a loop of excitation, and this depends on the fact. And then I'm going to go into a bit of the details on the fact that these kinase receptors, they have a very specific way of integrating excitability. So you add up excitation onto excitation, etc., then it triggers a very strong depolarizing effect and discharge. So it's really, I mean, as I said, this is the physiological mechanisms. Valérie and myself, we think we understand it quite well. It's not, yeah.
And then there is a last question before releasing everyone for
Fanny?
lunch. Yes.
Fanny, sorry, there are another question from Professor Philippe Vernier that was also related to the question asked by Anne Gelly about the immune response. And he was asking if there were, it will be only stereotactic surgery that will be intended with the product.
So, not shown here, and these are projects that we are working on in parallel, and you see on the screen with Dick Grimm, virologist from Heidelberg University. We are trying to two things. We have developed viral vectors which seem not to infect immune cells at all. So we have done a screening of new AAV vectors, which are on one point very strongly selective for DG cells and not other neurons in the hippocampus, but also they completely miss infection of immune cells. So that's a very nice point. And at the same time, we're trying to have viral vectors that we could deliver by not locally, but by retroorbital injections in the brain. But we did not find any vectors which could be completely selective for the G cells or hippocampal neurons, and that's a possibility. But for the moment, I mean, the target is to go for local delivery, and I think we have the chance in temporal epilepsy that this is done anyway by the surgeon.
Thank you so much. And then I may ask a last question from Luc Pénicaud, who is asking what is the mode of action of pilocarpine and what is the model, the pharmaceutical or other, in non-human primates?
So the action of pilocarpine it creates a major storm, electrical storm in the brain, and which is maybe also so it is an acute seizure which is major, and then you stop the seizure and then the animal can be normal for a few days, etc., and then progressively develops this seizure. So I mean, the pilocarpine is just here to create a major acute seizure event, and it can be kainate, it can be other ones to start with, and then the circuits reorganize and develop into epileptic circuits. And the other question was, sorry?
If there is what is the model in non-human primates, if there is a model on non-human primates?
I'm not sure that there are. I mean, we have been using normal primates to see the delivery aspect of the vector, to see how much we needed, how far it went outside of the hippocampus, etc., and whether there were expression in the liver, etc. But it was not a model of temporal lobe epilepsy. It was just to see the distribution of the vector once infected, transduced in the hippocampus locally.
Okay.
And I don't know of any model at the moment that can be used.
Thank you so much. But then maybe we can. I don't see other questions in the chat. We are also up to the time, so we can thank really Dr. Mulle for the great talk, and I mean it was really interesting, and thank you for accepting to participate to the first webinar of the series. And maybe to conclude, we can also say that this is just the first one, but we will organize a webinar every first Thursday of the month at the same time, and so we will communicate, of course, very soon what is the program for the next ones. And then with that, we wish you a great day, and we hope that we will meet each other at the next webinars, and maybe also in person very soon.
Thank you very much for the invitation.
Thank you very much.
Thank
Have a nice
you.
lunch and a nice day.
Have a nice day.
Thank you. Bye.